Hello, my name is Ian Wolfe. I'm an undergrad student here at Cal U. I'm a senior and I'm a biology major with a concentration in pre-med. Today, I'm going to be talking about metal resistance bacteria in acid mine drainage. To start off, I'd like to talk about what is acid mine drainage. And acid mine drainage is simply just water runoff from abandoned mines. In the United States, there's over 11,000 abandoned mines with high concentrations on the West Coast and on the East Coast. There's not many of them in the Midwest. And the acid in the mine drainage comes from pyrite oxidation, which results in sulfuric acid. The acid mine drainage also has high metal and sulfate concentrations. There's also another kind of mine drainage called circumneutral mine drainage, which has a neutral pH. There's lower sulfate concentrations, but there is a lot of iron contamination in it. And acid mine drainage is a major problem because there's no way to completely stop it. It can only be dealt with, and that can be done in two ways. There's passive treatment and active treatment. The passive treatment has a much lower cost. There's very little maintenance to it. However, over time, it can become less effective. Active treatment, on the other hand, is more expensive. There's a lot of maintenance to it, but it does work a lot faster. And the acid mine drainage pollutes many watersheds all over the country, affecting agriculture, drinking water, habitats, and recreation. As you can see in this picture, it just looks pretty nasty. So for the bacteria to live here, they have to be able to withstand low pH since it is acidic, making them acidophiles. And there's also a lot of metal in the acid mine drainage, so which suggests that they must be resistant to metal as well. There's a lot of copper, zinc, and aluminum in the mine drainage. I formed a hypothesis that the strain I got was resistant towards copper. To test this, a sample was taken from Boyce Park, which is near Plum, Pennsylvania. It was grown on a plate, and then from that plate, an isolate was purified using the streak plate technique. Once isolated, the DNA was extracted and sent out for shotgun sequencing. Shotgun sequencing is pretty much just breaking the entire genome into tiny fragments, making it easy to deal with. Once they're in tiny fragments, they're sequenced by themselves, like I said, since they're easier to deal with. Once those are all sequenced, they're ran through a computer, which overlaps the sequences like a puzzle. Once the sequences are overlapped, it puts it back together, giving us the entire genome. Once we had the entire genome, it was annotated, and during annotation, we looked for certain genes. The genes I found suggested that it was resistant towards copper, and then that hypothesis was then tested. This was done using a disk assay. A disk assay is where you streak a plate with bacteria all over, and then you place these little discs on it, which have whatever you'd like to test resistance towards, whether it be metal or antibiotics or what have you. In my case, it was copper, so these little discs had concentrations of copper on them. Once they were placed on it, the bacteria was let to incubate for 48 hours at 30 degrees Celsius. This was done with low concentrations of copper and high concentrations of copper to see how resistant the bacteria was. Like I said, during annotation, I found many genes suggesting copper resistance. There was also one of them which suggested arsenic resistance, but I only tested copper resistance in my experiment. The isolate is also very probable to be novel because the closest related bacterium shares 89% of the genome, and that just shows that it's very probable to be a novel bacteria. But to figure this out, there'd have to be confirmation studies done. So here's my positive control for the disk assay. It had no discs on it, which is just to show that the bacteria was able to grow in the conditions given. And over here, we have the ones with the low concentrations. It was done on two plates. The concentrations for this disc and this discs were the exact same. It was just done twice to make sure there wasn't a fluke and the bacteria was able to grow on both of them. And these plates have bacteria all the way up to the discs, which have copper on them, which shows that the bacteria is just completely resistant towards the copper. If the bacteria was not resistant, then there would be a zone of inhibition here, as shown in the previous slide over here. There would be a large gap. And as expected, on a high concentration of copper, the bacteria also grew right up to the plates. It's hard to tell that there's growth on them, but there is growth on the plates. So in conclusion, I found the bacteria in the acid mine drainage that I got was resistant towards metal. And this just shows that the bacteria are adapting to live in the acid mine drainage since it's very acidic and has high metal concentrations. And knowing that this bacteria is resistant towards copper and maybe other metals, it could be the key to bioremediation. Maybe not single-handedly, but with other studies done on the bacteria and the mine drainage, it could lead to better ways for bioremediations. If I were to continue this experiment, I would do confirmation studies on the bacteria to see if it is novel. I do plan on doing this next semester for my research project, and that's where this project would go. Thank you.